So today's seminar is on roots for drought overcoming the root phenotyping bottleneck in cereals, presented by Professor Daniel Rodriguez from the Center for Crop Science. Hi everyone, I'm Dongxue Zhao, and I'm an early career research fellow from Center for Crop Science. And next slide. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. This seminar is scheduled for 12 o'clock until 1 p.m. And at the end of the seminar, we will hold a Q&A section. And if you have questions, please use the Q&A button at the bottom of the screen, not the chat box. And we will address them at the end of the seminar. So Professor Daniel Rodriguez was trained at Wageningen University as a biophysicist and worked in crop ecophysiology and systems modeling. He is presently editor-in-chief of Sustainable Agriculture and lead projects in crop ecophysiology, systems modeling, adaptation to climate variability and change, and sustainable agriculture. Daniel, floor is yours.
Thanks, Dongxue. Can you see all right my screen?
Yes.
Okay. So the title, as Dongxue said, is Roots for Drought, Overcoming the Root Phenotyping Bottleneck in Cereals. And this is a contribution from several people, Dongxue, Peter de Voyle, Farsinet, and Sebastian. Let's see how we go. So the origin of this presentation is a new project and some existing activities that we have been running for the last year or two on phenotyping, high throughput phenotyping of rooting systems. The new project activity is funding from the GRDC. It's a large collaboration between Queensland, QAAFI, CSIRO in Canberra, the DPIRD in Western Australia, and IPK, which is an institute that specializes on roots in Germany. And the project is in collaboration with several companies, breeding companies for wheat and for sorghum, as well as with providers of digital ag services and precision agriculture. As you can see, the team is vast and involves people from multiple disciplines, from modeling to physiology to pre-breeding to statistics, and in both wheat and sorghum, and as well as imagery analysis, like the work from Andres, and 4 PhD students, 3 in Australia and 1 in Germany. The value addition from CSIRO is on new environments and new techniques to quantify rooting systems in the field. And Hannah Schneider from IPK is a specialist in root anatomy and the study of plasticity in roots and crops. What we expect to achieve from this project is very much an accurate, fast and cost-effective high throughput phenotyping tool for impactful root traits that affect yield stability. And you can see there are a number of words, the terms are underlined. That is just that we really mean to be impactful. There is also additional objectives, which is going to be to quantify the genetic diversity in sorghum and wheat on some of these root traits. And we want to answer a key question, and what is the value of including root traits in breeding programs across different target production environments in wheat and sorghum? And in the meantime, just to also revise the functions and the way that we are using to simulate root growth and function is in our main model. So as you can see, it's a very vast and ambitious type of project. So why do we need to phenotype for roots? Well, so far phenotyping for breeding and agronomy has been conducted primarily above ground. And so we have used many different techniques, and Andres is a specialist on that. But so far we have sort of ignored what happens below ground. And that brings a lot of questions, and what is the value of root traits just to include in breeding and agronomy programs? And we don't really know a lot about that. Most of the phenotyping that has been done so far has been primarily what we call structural phenotyping, and tends to focus on the form or architecture of the rooting system, and it has been done using different techniques, using pots, using growing plants in chambers. Let me see if I can get a laser pointer. Oh, it's fancy. In chambers, or just going into the field and extracting soil cores and counting roots, as you can see here. This is in wheat and in sorghum, very, very similar approaches. Though there is a bit of a big gap in what we think about structure of form and function. And that can be simply seen in when we are looking into what happens with root water uptake in the field. For example, here we have roots. It's an example that we have roots growing up to 55 centimeters in depth. So this is the water extraction from different soil layers that you can see here. And the roots are at about 55 centimeters, but then there is a rainfall event. And for about a week, or a bit more than a week, there is no water use from the deep layers, even though there are roots in the deeper layers. So this is something very, very common that we and farmers observe when we install these new probes and new technologies that allow us to continuously monitor soil water. And these are things that are quite new to some people and completely ignored in models. And it's like the crop is just saving the deep water for later. So it's transferring water use from the deeper layers to later in the season. And when you have environments where you have frequent rainfall events, that tends to be exacerbated and is completely ignored in many analyses. So even though there are roots, those roots might not be taking water, and that is something that has been a bit of an issue when we just focus on the form of the rooting system. So what is the alternative? It's just to look at the function, or just to complement the studies that have been looking into structural phenotyping with studies that are looking into functional phenotyping, and see how the rooting system works. And to do that, we have developed some new techniques that they use an electromagnetic induction sensor that goes on top of the soil. And very much we can say how much water is in the soil profile, down, in this case, down to 3 meters. And that machine is just driven inside the crops at different stages, different windows. For example, if we do that around flowering, in a window of about 10 days, for example, or a week, and we can slice the water and water use from different layers. So then what we can look at is how deep are the roots extracting water and what is the proportion of water being taken from different layers. And that is what is done by using the pipeline that was developed by Dong Shu, where the values of the sensor are calibrated for soil moisture to predict moisture content at different soil layers and crop water use when you pass the sensor in a window of about a week or 10 days, for example. So it's very, very simple. So we infer on function by just looking into water use in this case. The next step into this technology is the root bot that is coming now in August. That is a new facility in QAAFI that is going to allow us to run this in an autonomous sort of robot that is going to go on top of the crop, because this is a high clearance vehicle, and is going to also allow us to take soil cores and validate the results for moisture and roots. How does it work? Well, very much from those, what you get from this technology is, if you see, this is a very large experiment that is 380 meters long and 82 meters wide and 1.5 meters deep. So it's a very big chunk of soil. And we calculate an index like that is, we call it the root activity factor, that is related to water use for each individual layer relative to the initial soil water and the size of the crop. We can clearly see that this is very much a slice of the big cube just for one side, where we have different reps and different treatments, but very, very clearly shows that there is a relationship with, for example, plant density. So where there is more higher plant densities, there is soil is being drier, and when you have lower plant densities, the soil is being a little bit wetter, or there is less water activity just because of the demand. So this is at 7 leaves, and that is the demand driven. But also you can see that there are all the treatments and can very much represent what is happening in the field. And what is most interesting is that these values of root activity have been directly related to root length density. So what we did is just we went into each of these plots in the fields and took a soil core and washed out the roots and measured how many centimeters of roots we had per cubic centimeter of soil. And it's interesting to see that the EMI derived index can really very closely predict what is the root length density as you go from deeper, shallower layers down to deeper layers. So what we are seeing here is very much related to where the roots are. So that can be used in different ways. And one of the ways is to look at, okay, we want to know what is the genetic and the management variability derived from these EMI root traits. And we would like to know what is the association of these traits with yield components, yield quality, yield stability. And that is probably going to be the main focus of the talk today. But also this offers us a unique opportunity to revise how we are presently modeling root growth and function in AppSim. That's at the moment is using a very, very simple approach. And then we have other interests. So is there any value in selecting for what root traits in what environments? And that is something that the breeders in the team are asking and are very interested. And also related to the agronomy applications and precision agriculture applications. So where is the value? So if we are able to map maximum rooting depth across fields, that can be extremely valuable to map, for example, plant available water across large areas of fields. So in this presentation, I'll talk a little bit about two important traits for crops, and one is to have stiff and deep roots that they can access water deeper in the soil layers. And if you can imagine that we have conducting this survey with this machine at 2 times, about 10 days apart, and we don't have any ingress of water through rainfall or irrigation. The only active source is going to be the water extraction, and that is going to allow us just to derive some indices of, for example, what is the root activity shallow relative to the total, or what is the maximum rooting depth. Just a simple example is one of the 2 indices that we are also using. And this is associated to Dong Xu's Australian Research Council project with Pioneer. So in simple terms, this comes from a trial in Brookstead in the field, in the farmer's field, and using 20 sorghum hybrids of contrasting stay grain. Some of those are commercials and some of those are experimentals from Pioneer. And this is a very simple way of showing what is the relationship between hybrids that had high yield, but also high test weights relative to, for example, the rooting depths of flowering. As you can see, there is no association at all. So you have hybrids that had high yield and high quality at different values of rooting depths of flowering. But the first thing we see then is that, okay, roots keep on growing from flowering to maturity, and they can grow for up to another 30 centimeters in depth. And what is interesting is some of those materials, they have differential capacity to grow deeper roots. And if you are generous with me, you're going to see that there is more red on this side of the graph on materials that had a higher rate of root growth, or a higher root growth between flowering and maturity. And in the same way that this is the root activity factor. So small values of this root activity mean that there is more water being used from deeper layers, and a higher value of this index indicates that there is more water used from shallower layers. And again, if you are generous with me, you're going to see that there is more red on this side of the graph than in this other end of the graph. So, and that is giving us some indication that these indices that we are deriving, they can have some interesting relationship or functional relationship with grain yield. So are these materials having different root angles? So we also did some analysis on novel root angles for all these 20 materials. And we do see that they do differ quite a bit, even though many of these are commercials and as well as experimentals. But what is the relationship to yield or important traits? So what we can see is that the average root angle, which is the average between the left and the right angles here in these trials run on a chamber, have been related to the grain yield, saying that the narrower angles in this particular trial, let's remember this is on one particular environment only, and the trial has been repeated this year, and we still haven't crunched the numbers. We have another 2 environments. And So we're looking forward to see if we can replicate these results. But also we can see that the rooting depth at flowering was also related to an index of a stay green that was derived from imagery during grain filling. So there seems to be some indication that root angles and rooting depth and these indices derive from our electromagnetic induction methods are telling us something about the capacity of the rooting systems just to go deeper. And if you see in a bit more detail, so you can see here we have the yields in this biplot, and obviously going in an opposite direction to the average root angle. And higher yields also are associated to the deeper rooting depths and maturity, deeper water use from deeper layers at flowering, and higher total water use. So some of these indices obviously are telling us something about what is happening with these traits. So narrower root angles and show deeper rooting systems with larger values of water use between flowering and maturity were associated also to higher yields. And this is not new to some of those growing, working with sorghum, but this is probably the first time that we can see a direct relationship between root angles and yields in the field. So I said I was talking about steep and deep, but I'm going to add another word that is plastic. And that relates to traits, for example, that they are stable across environments versus traits that can be plastic or changing across different environments. So you can have a trait that is plastic, and that phenotypic plasticity is resulting from complex relationships between individuals, genotypes, the environment, and the management. But also you can have hybrids that are more or traits that are more or less plastic. And also you can have traits that they show interactions. And this is not new. Even Darwin was criticized because someone was saying that whatever you are looking at, what you are seeing, these phenotypic differences is not different species, it's just phenotypic plasticity. So at that time, the concept of phenotypic plasticity was already there. At the moment, what we are using is we are quantifying plasticity using what we call the slope of the reaction norm, which are these slopes. And we calculate that under specific influences, genotype by environment, management by environment. What we find is that stable traits tend to be associated to traits that are plastic. So for the trait to be stable, something else is going to have to give up when you have different environmental signals. And that means that there is a hierarchy of plasticities in crops. And there is also what we call specific adaptations. For example, this material appears to be specifically adapted to these good environments. But also there can be a cost. This is going to have a cost. It's doing it very well in poor environments, but in bad environments, but in good environments, is losing yield to this one. And that means that there might be effects of maladaptation that need to be taken into account. What we do know is that these effects are under their own genetic control and therefore being able just to be manipulated to increase fitness. Just to look at the concept of plasticity, I'm going to look into a data set of 32 environments, 8 commercial sorghum hybrids and 4 plant densities. And we use these as the source of environmental stress. And you can see that the yields were extremely variable across environments and across the treatments. For in this case, we even have 3 sowing times, an early and a spring and a sorghum. And that very much changed the patterns of stress. Those early sown, they had a higher frequency of stress types 1 and 2, We can see those stress 1 and 2 are just little stress or little late stress. And those late plantings, they had a higher frequency of more severe stress, which is defined in this way by the ratio between saltwater supply and saltwater demand. For those that have heard Graham Hammer in the past, so they know what we are talking about here. And then we are going to be looking at these different responses of these different materials across these highly contrasting environments. As you can see, we have a lot of interactions here. Some materials are doing very well in good environments. Some materials are not doing so well in good environments. So let's see the hierarchy of plasticities in sorghum, which is something that hasn't been published before. This is something new. Though it's suspected that the highest plasticity, the highest values of plasticity are going to be on grain number components compared to grain weight components. And here we have, for example, this is total grain number, mine stem grain number, total grain yield, tiller grain number, and on the other side, on the other end, so we have more of the grain weights, the main stem grain weight, the tiller grain weight. And the root traits that we measure in this case that are the maximum rooting depth and the root activity factor are sort of about in the middle. So some traits, they have more plasticity, and some traits, they show less plasticity. And what we conclude so far is that grain number components tend to be more plastic, and the grain weight components seem to be more conservative. And there have been theories about evolutionary theories about why this is like that. And in this case, root activity, and maximum rooting depth, they show about intermediate values of plasticity. So let's see how this is useful to explain our results. So we have this conceptual framework, which is you have more plant available water or less plant available water, and we have hybrids that are more responsive, hybrids that are less responsive. And that is very much coming from the data sets that we have over there. So if we look at all the traits, we can look at, for example, the root activity. So as I said before, we can postulate that the hybrid A, that or the hybrid B, let's say, that it has a high capacity to take water from deeper layers, is going to be better suited to these drier environments. However, the hybrid A is very stable in this trait and is not able just to compensate or to modify the rooting system to access water in these environments, but is doing really very well in very good environments. In the same way, you can say that the other trait that we have of interest is that we're going to have deeper roots in some materials and shallower roots in the other one. So these are very much the hypotheses that can sort of explain these results of variations in plasticity to yield. So what we have found so far is that the relationship between the trait plasticity and yield components. For example, here we have grain plasticity on grain number, and that means these are the different 8 materials, and this is the capacity of materials to maintain a stable grain number across very variable levels of plant available water. And that has been highly related to the plasticity, highly related. This is a 0.078, which is significant 10%, and negatively related to root plasticity, root activity plasticity. And what does it mean? Is that if you have a high plasticity in root activity, that means that these materials are going to be able just to change and take water from deeper layers as soon as the deeper layers are drier, and that is going to stabilize grain numbers. the same for grain weight. In this case, relationship is significant, 5%. We can see that this is rooting depth plasticity, and that is the capacity of different materials, of these materials here, to put deeper roots as soon as the environment starts to get drier. And what we can see is that higher root depth plasticity, or the capacity to put roots deeper, was related to stabilizing grain weights. And the opposite, to those materials that were not able just to modify the rooting depth, showed a bit more variable grain weights between drier and wetter environments. And this article has been under review at the moment, and let's hope that the reviewers find it interesting. So, and that calls for a number of points, and one is how we are simulating this root growth and water uptake in upstream. Obviously, there are many things that we are completely missing at the moment. This is a simulation of upstream that shows very much the root growth in depth and the accumulation, the value of water use and soil evaporation, and what is happening at the same time with water use. So, and when you compare this, when we see in reality, obviously the model is not able just to reproduce this transfer of water for later in the season. And we do have some hypotheses that we are planning to test with our group in IPK in Germany, and very much relates to the hydraulic conductivity of the rooting system at different soil depths. That could probably explain that. Though when we introduce that into upstream and start to play to see what happens with upstream presently, and what happens if we introduce these delay the water use from deeper layer, we didn't see a lot of variation. So, but this is very, very early stages. So we are just starting to look into this, and we are hoping to be able to progress a bit with a collaboration with Germany. So some take-home messages. So in addition to characterizing the mean value of traits, now we think obviously that we need to characterize also their plasticity. So this is something that we haven't been able to do in the past, or we haven't really thought much in the past. And now we have a high throughput functional root phenotyping capacity just to do so, just to monitor root traits and their plasticity. And that complements the structural root phenotyping approaches that have been very much championed by pre-breeders and in QAAFI. Though very little is known about the costs of plasticity, so that wedge over there to see what happens with this material, why this doesn't have the fitness in good environments as well. Is there a cost by putting these roots deeper, to reacting to the environment and allowing the crop just to sustain growth in drier conditions? So, well, those things are part of some of the questions that we are also going to be exploring with the group in Germany. And in addition to that, the group in Germany, which is Hannah Schneider, is going to contribute significantly to changes in root anatomy and the plasticity and genetic differences across the materials they're going to be testing in wheat and sorghum, which is going to be very exciting. And always with the modeling root growth and function focus to try to see how we can better represent genetic responses to management and environment. And that is very much work in progress. So, and that is all I have to present, Dongshu. So I'm happy to take on questions if you want.
Okay, thank you very much, Daniel, for the great presentation. And this is the Q&A.
Oh, and I had my camera off. Look at that. Didn't realize, sorry.
Okay. So now, if you have any questions, please feel free to put into the Q&A box. And before I'm asking the questions from the Q&A, I have one question.
Yes.
Yeah, it's of course understanding the plant trees variation and also the plastic responses of the both structural and functional trees under variable environments is very important, is critical for predicting and managing the effects of climate change on the crops. And so what are the new opportunities you think the new opportunities of this in-field high-throughput root phenotyping method bringing to us in the breeding or pre-breeding programs in agronomy and crop modeling?
If I understood well, the phenotyping of novel root angles that has been pioneering in QAAFI for some time already, in the past has been difficult to relate to growth and yield traits. In this particular trial that we had, it was actually a perfect trial just to quantify the influence of root angles on yield, and novel root angles on yield. In that trial, it rained until flowering, and from flowering to maturity, it didn't rain at all. And we focused the analysis between flowering and maturity, and that is very much, you would say, a perfect trial to do that. When you have a perfect trial, things are easy just to identify these signals. But still, what we are doing is just that root angle is a static trait. Okay, it's just you can say, okay, this cultivar has this number or this angle. But what you cannot say with those techniques is how variable is that angle when you are exposing that cultivar to a particular stress. So what we are doing at the moment is we are changing the root chamber novel root angle protocols to see if we can at the same time calculate plasticities in root angles, and if those plasticities in root angles are related to any plasticities later in the season on water use and root growth. So we, at the moment, there is a trial that Fascinet is running, and obviously on a limited number of genotypes, on those very same 20 experimental and commercial hybrids. But as a test, if it goes well, we might want to scale to a larger number of materials. But yeah, there is a good approach to quantify novel root angles using those chambers. We want to see how we can use those chambers also to calculate the plasticities on those materials, and to see if there are any variability on plasticity in novel root angle across the existing germplasm that we have in the wheat and sorghum programs.
Okay, thank you. It's quite interesting to incorporate both the root phenotyping in the chambers together with the root phenotyping in the field. So we have several questions come into the Q&A box already. I'm not going to read them in order. I just now is the question from John Dixon come to my eye. So very interesting results, especially here ranges of root plasticity and the potential improvements to epicene should be valuable. So the first question from John is, in relation to the 6 environments on the first experiment, please comment on differences in results for different types of soils. And the second question is, how could farmers use EMI for routine management? How soon might be the EMI technology be cost effective for farmer adaptation?
Well, the 36, I think it was 36 or 32 environments that we had in the large data set, obviously they differ on soils as well. And very much the only way that we are not looking this from a soil scientist perspective, but we are looking that from the plant and how the plant react to those environments. So the different sites that ranged from the Liverpool Plains up to Emerald, going through as west as Surat. So those environments are captured by a number of covariates, and those covariates are primarily related to environmental conditions, to climate conditions, and soil conditions such as plant available water. So any other effect is it is part of the variability that we have in that x-axis in the plot of environmental yield and treatment yield. How the technologies are going to be used for industry. Mm-hmm. Well, industry are involved in this, and they are part of the project, and they already come with ideas on how to sell these products. So farmers are not going to be doing this. This is going to be done by a service provider like Airborn Insight or DataFarming, and we are, they are part of a project, and we are discussing what applications they would like to develop from that. So that I think is bit early stages, but there are a number of ideas. So mapping plant available water across fields, mapping maximum rooting depths, so identifying subsoil constraints. So it is, it is quite diverse. And for the breeders, they are primarily interested to know, okay, which ones of my materials I can promote as a material that has a particular rooting system that is better suited to a particular environment. There is value for everybody, and that is why they are part of it.
Okay, thank you, Daniel. I would like to add a little bit more to the first question about the 6 environments, the soil types effect. So at that 6 environments for root plasticity calculations, it's very much the same soil type across that 6 environments. And this heavy clay soils is the black water source, and it's with a clay content more than 50%. So we all know that the EMI technology works better on highly clay soils. So we haven't tested this technology on sandy soils. And in our new GRDC project, because we have collaborators from Western Australia, from Canberra, and we will test in our new project, test the technology performance on different soil types. And yeah, to see how it works and work on how to improve the performance of the technology, maybe incorporating different geophysical sensors together to work on sandy soils instead of just rely on EMI. Yeah, it's
okay. Yeah, we needed an answer from a soil scientist.
Yes, exactly. And there is another question from Ribbit Core. Sorry if I pronounce your name wrong. In case of weight, is there any relation between root plasticity at the tillering stage?
We are very new to doing this on wheat, and the trials, all the data I presented is on sorghum, and we don't have at the moment a trial on wheat. Actually, tomorrow we are planting, and I should be at the moment marking plots in the field here. So if you ask me that question next year, I'll be happy to answer.
Okay, thank you. There's another question from Helen. Thank you for your invaluable presentation. I have a question regarding the diverse responses of different genotypes under stress conditions. Some genotypes exhibit low plasticity, maintaining consistent performance regardless of variations in growth conditions. While this trait demonstrates commendable resilience, it often results in poor production yield. On the other hand, certain genotypes display high plasticity in growth, adapting well to diverse conditions and achieving high production levels. Could you please provide an explanation for these contrasting responses?
What I think is that is what we are trying to do. And what is important to notice is that so far we have used a very limited germplasm pool. And the idea is that in this coming season, we are going to test the technologies on populations that are known to have contrasting root angles, though the plasticity analysis is pretty new. There is no reference on plasticity on these traits or their relationship to grain yield or grain yield components. So I think this work is it is new, and the results are quite innovative in that sense. But there are at the moment I think we have more questions than answers, and why a material is having a different reaction and being able to put roots deeper compared to another one, I have no idea. That is another question that we might want. Now we are having 4 PhD students. Hopefully some of those students are going to be focusing on some of those questions, but they are all relevant questions. It is exciting just to be able to do this, and I think at the moment, as I said, we have more questions than answers. And what we do need is a better understanding of the biology, so that the biology that can be summarized into models in a more realistic way, so that then our models, they can be used to really characterize phenotypic responses from different genotypes. That at the moment, I would say that it's been focused on above ground traits, ignoring what happens in the rooting system. And what we show is that, well, we need to look at the rooting system as well.
Okay, thank you. And I think we have more time for one more question. Is from Professor Michael Uwadi. When you compare root function plasticity between genotypes, do you take account of actual versus measured soil water, or do you assume each genotype has the same soil water availability in adjacent plots?
So these, depending on what index, the trials are run on fields that are considered to be uniform, and we run surveys to make sure that the trials, the soil water content is uniform in the field, and that is one point. The other point is that the root activity factor, it factors in the soil water at the start of the period that is that the trait is measured. So not only that, but also the canopy of the size of the canopy. So they are the two main confounding factors that you could expect, or at least the most important ones. And those two factors are included in the indices that we are producing and that Dongshu published this year and last year. Yeah, but happy to talk to you, Michael, and have another chat. Actually, we are doing this also on pulses. We have 2 years of data on chickpeas, but we have been a bit slow at crunching those numbers. So we are completely flat out. So happy to have a chat and show you the results, and we can brainstorm something.
Yeah, okay. Thank you, Daniel. I think it's very much we have done today. And we go to the next slide.
There is no respiration. What is the next slide? Sorry. Okay. I see that Michael had more questions, so Michael, happy to follow that up. And
yes,
also
sorry. Yeah, if someone's question I haven't got time to read, please feel free to send us emails and to follow up questions, because we have time limitations and we have another 3 slides too.
Yeah. Also, Alan had a good question, and Samir, and yeah. So let me know when you want to have a chat, and we can have a coffee and discuss all this.
Yep.
So you want the next one? Yes.
Yes. Okay. So the next science seminar will be on 28 May at the same time as today, and will be presented by Dr. Bradley Campbell from the Center for Horticultural Science. And the seminar title is Improving the Impact from Current and Future Genetic and Genomic Investments. Next.
My computer says no. Oh, there you go.
Okay. And if you haven't already signed up to the seminar invitation list, please scan the QR code and sign in today. And thank you very much for attending our seminar today. Thank you very much. Thank you, Daniel.
Well, thank you.
Yep.
Yeah.